Hello, class. Professor Fisher. Now we're going to go into neuron synapses. This is where we have the connection between neuron and its target, which could be another neuron. And so basically we know how the signal is sort of propagated along the neuron, but then how is that signal transferred to the next neuron? Right, neurons must communicate with other cells. Very simply put, neurons have to communicate with whatever, they have to send a signal to the brain and communicate with the neurons, process. They send signals to muscles, they send signals to organs. That's how neurons carry out their function. A synapse, the term synapse, is where a neuron meets its target cell. Right? It can be electrical or chemical. Right? Now, when we say electrical or chemical, you may say, well, what about mechanical? What about muscles? The synapse, the connection is electrical or chemical, but that doesn't mean it can't result in a mechanical response. Okay? Neuronal synapses can occur between an axon of one neuron and another part of another neuron. So it doesn't necessarily have to be axon to axon. You'll see. We have different examples. All right, so we have axodendritic, which makes a lot of sense. Axon of one neuron to the dendrite of another. Remember, the dendrites of the neuron is where a neuron usually receives a signal. But we do have axosomatic, axon of one neuron to the cell body of another neuron. That can happen as well. We have axonic synapses between the axon of one neuron and connecting to the axon of another neuron. Okay, so these are all ways that neurons can communicate with other neurons. Terms used to describe neurons, we have presynaptic neuron, that's the neuron sending the signal, and the postsynaptic neuron is the one receiving the message from the presynaptic neuron. Synaptic transmissions, all right, let's go over those. So we have a transfer of chemical or electrical. You may say, well, we haven't really talked about chemical. We've only talked about really the electrical, the electrical charge going through. But you'll see that's, remember, these are well, you'll see. It can be chemical or electrical, not just the electrical that we've talked about, between neurons at the synapse. Right? This allows for things like movement, cognition, thinking, sensation, emotions. The average presynaptic, the one sending the signal, neuron, forms synapses with about 1,000 postsynaptic neurons. Right? And then postsynaptic neurons can have as many as 10,000 synaptic connections. Right? So this is it. There's a lot of connections. In life, let me just say this, if we would be wise to model our lives after our bodies. Your body, everything works together. Everything works together. And they say in life, we say, it's not about what you know, but who you know. That's basically how our bodies are set up. It's not, everything, the neurons will connect and send signals with, they transfer information. No part of our body, not that I can think of off the top of my head, is really isolated in how it works. It all works together, all connected. Everything has its own, every part of your body has its strength. So even the smallest neuron is connected to like 1,000 other neurons, so it can communicate and send signals. And that's one of the things that makes our body so amazing, is how interconnected it is, how everything plays its role, has its part. All right. Electrical synapses. Okay, so electrical synapses occur between electrically coupled gap junctions. This is similar to what we've been talking about so far. Axial lemmas of each cell and synapse are nearly touching, gap junctions align, channels forming pores that ions of the small substances can flow through. We find these in the brain, programmed automatic behaviors such as breathing. We also find this in cardiac and visceral smooth muscle, allow for coordinated muscle activity. Electrical current can flow directly from axoplasm. Remember, that's the cytoplasm of the neuron, of one neuron, and creates two unique features. Transmission is bidirectional, can go in either direction. So either neuron could be pre or postsynaptic, depending on the direction of the flow. And transmission is nearly instantaneous. So less than 0.1 milliseconds. That's very fast, much faster than the chemical synapses we're going to talk about, which are 1 or more milliseconds. All right, so basically electrical synapses, the idea here is that the same ions that depolarize the last section of one axon flow through these ion channels, these gap junction ion channels, and go right on to depolarizing the next section, the new neuron at the synapse. But we also have chemical synapses. So electrical synapses, we just basically depolarize one axon, and then the ions flow right through and just continue depolarizing, so the electrical signal just keeps right on flowing, extremely fast. But we also have chemical synapses, right? Not necessarily based on the same type of setup, a little different setup. They make up the majority of the synapses in the nervous system. They're not as quick, more efficient but they're more efficient than electrical synapses. Convert electrical signals into a chemical signal. No strength is lost, as the electrical synapses are. All right, so what happens in a chemical, this more common chemical, and how does that compare to an electrical synapse? All right, so in the chemical, we have these what we call synaptic vesicles. They're filled with these chemical messengers called neurotransmitters. You probably have heard of some of these. We're going to talk about them. We have the synaptic cleft, this extracellular fluid-filled space, which separates the presynaptic and postsynaptic. So in this case, the presynaptic and postsynaptic neurons aren't joined together with just by this gap junction. They're actually slightly separated, right? We have this which we have the synaptic cleft. The postsynaptic neuron has neurotransmitter receptors. We have this little synaptic delay. So there's going to be a little bit of a time gap. Remember, these take a little bit longer than your electrical synapses. And chemical synapses are unidirectional. So they can't go both directions. One neuron's always going to be the presynaptic, one will always be the postsynaptic. But that allows for variable signal intensities. You can get much stronger responses in the chemical synapses. All right, so you have at the end of the axon, you have these neurotransmitters, which get released into this synaptic cleft, this gap between the axon of one neuron and the part of the other neuron that you're connecting to, whether it's the dendrite, the cell body, or the axon. And this is what happens. So action potential, presynaptic neuron triggers opening of voltage-gated calcium ion channels. Now, normally we've talked about potassium, sodium, and now we have calcium. Influx of calcium ions causes synaptic vesicles to release neurotransmitter into synaptic cleft. Neurotransmitters bind to receptor postsynaptic neuron, bind to the receptors, ion channels open, leading to a local potential, possibly an action potential if threshold is reached. What's that threshold? Negative 55 millivolts. All right, so here we have at the end of the axon, calcium gated channels allow for this neurotransmitter to release its vesicles into the synaptic cleft, bind to the ligand gated channels here, allow for ions to flow in. If you get that action potential, that negative 55, we get to at least the threshold, then we'll have that action potential will spread through that next neuron. This is a video, you can watch it here. All right, we have this term called neural integration. So when neurons receive input, inhibitory, excitatory, some telling them to stop, some telling them to go, from multiple neurons. We have this thing we call neural integration, which is a process in which postsynaptic neurons integrates all incoming information into single effect. So remember, neurons are connected to thousands of other neurons. So I'm getting all this information at once. All right, let me take the positive, the negatives, okay, add it all together. It's mostly positive. I'm gonna send the signal. Or if I take the positive, negative, add it all together and it's mostly negative, I'm gonna stop sending the signal. It's telling me to stop. So all input from several postsynaptic potentials added together to affect the membrane potential. All right, so if all the signals that I'm getting, a threshold will be reached if the sum of the excited signals, the ones telling me to go, are enough to overcome the ones telling me to stop. Very simply put. All right, so that's basically the diagram showing you that. I don't think I've ever put that on a test before, actually. I had another video in here you can watch. Some more slides for more information. All right, some different neurotransmitters. Let's talk about those. Nearly all neurotransmitters undergo similar pattern of use. Despite that there are over 100. They're made in the cell body or the axon terminal, packaged into synaptic vesicles, right, in the cell, released from the axon terminals of presynaptic neurons. They cross that synaptic cleft, bind to the specific receptors. Effects are often rapidly terminated through removal or degradation. We have multiple types of neurotransmitters: tropic receptors. Forgive me for my pronunciations. I don't say these couple words very often. Components of ligand-gated ion channels directly control movements of ions into or out of the neuron when binding to neurotransmitter. We have metabotropic receptors within the plasma membrane. These are different types of neurotransmitter receptors associated with separate ion channels connected to metabolic processes initiated when neurotransmitter binds. Some more on metabotropic receptors. You have G proteins, group of intracellular enzymes associated with many metabotropic receptors. They create this like cascade of enzyme catalyzed reactions. Then you have some second messengers, open or close ion channels in postsynaptic membrane. Cyclic AMP, that's one that you'll probably see if you go into medical field. Common second messenger derived from ATP, multiple functions in the neurons. CAMP binds to group of enzymes, add phosphate group to ion channels, triggers channels to open or close. These are just diagrams of different ones of those. And how they work. Notice this, ligand binding to the site, opening up. And this metabotropic receptor, G protein inactive. Neurotransmitter binds here, G protein activates. And you have an enzymatic reaction, second messenger, allows for this ion channel to open. Major neurotransmitters, all right? So we really want you to understand. So binding of neurotransmitter to receptor leads to an excitatory effect or an inhibitory effect. Excitatory effect, one that wants to, it's like a yes, basically. Inhibitory effect is like a no. So you can get multiple, remember a neuron is connected to multiple neurons, thousands. And so you can get like 600 yeses and 400 no's, and okay, we're going to continue to send this signal. Most neurotransmitters can have both effects. Depends on which postsynaptic neuron receptor they bind. And single neurotransmitter may have several receptor types, right? So a neurotransmitter can cause a signal to, something to keep going, or a neurotransmitter can cause a signal, can be telling a signal to stop, right? So this is, and it'll make more sense when we're talking about different processes that are happening, and these different neurotransmitters and how this all works together, but you'll see. Major neurotransmitters are classified into 4 groups based on their structure. Now some of these we'll talk about a lot more details than others. Acetylcholine is one you're gonna hear about a lot. CNS, PNS, brain, spinal cord, muscles. Acetylcholine is so we're going to talk about that when we talk as we continue on in this nervous system. As we talk about the muscular system, we'll definitely see acetylcholine, but you'll see some of these other ones too. Serotonin is one you've probably heard of. Histamine is very much related to allergic responses. Opioids. Obviously that is and has played a major role in our news in the last, I would say, 10 years, especially in Philadelphia. Right? Major neurotransmitters, acetylcholine, a small molecule neurotransmitter widely used by the nervous system. Cholinergic synapses bind ACH, this is acetylcholine, in the neuromuscular junction, so nerve coming to a muscle, neuromuscular junction. Also within the brain and spinal cord, and within the autonomic nervous system. We're going to talk about that in the future. Largely excitatory, so wants to send the signal, it's a yes. Does exhibit some inhibitory effects. Normally this is something when you want to send the signal. Synthesized from choline, acetyl-CoA, don't really need to know that. And it also quickly degrades. After it sends the signal, it quickly degrades in a synaptic cleft. And then we have these biogenic amines, 5 neurotransmitters synthesized from amino acids throughout the CNS and PNS for regulation of homeostasis and cognition. All right, so we have norepinephrine. All these are going to be pretty important. Norepinephrine influences your heart rate, blood pressure. All right. mainly in autonomic nervous system. Epinephrine, similar function, used widely as a hormone by the endocrine system. Autonomic nervous system. Dopamine, used extensively by the CNS. We talk about, if you know of anybody who struggles with illegal drug use, dopamine is a huge deal. Movement, coordination, emotion, motivation, that's used in your brain oftentimes, and that's what helps give that feeling of high, that high feeling. And the release of dopamine is, and it, in conjunction with the use of opioids like heroin, can give this sensation, that people will literally kill themselves to try to feel. They say the feeling is just amazing, especially the first time. I don't recommend it because the addiction is terrible. It can lead to your demise. Serotonin, synthesized from the amino acid tryptophan. Most serotonin secreting neurons are in the brainstem. And we also have histamine, synthesized from the amino acid histidine, regulation of arousal and attention. There are some psychiatric disorders and treatments. Psychiatric disorders affect your thought, generally treated by modifying the synaptic transmission, how our neurons communicate, basically. Psychopharmacology, drugs that affect your brain functions, which can target either the action potential generation or some aspect of the neurotransmitter, which makes sense if we can control one of the two. Here are some illnesses that I'm sure some of you have heard of, familiar with, maybe even know somebody, or maybe have struggled with it yourselves, depending on. Schizophrenia, thought to result from excessive release of dopamine. People who this can be people who use drugs, unfortunately, can have this as a result. And so they get these psychotic episodes. Too much dopamine being released, which is released by certain drugs, can cause this release. We manage it by blocking the postsynaptic dopamine receptors. All right, so you don't really feel that great afterwards. They say you feel with the medication, you feel like basically sleepy and non-responsive. Depressive disorders, disturbances in the mood, thought to be a deficiency in transmission of serotonin, norepinephrine, or dopamine. So you don't have the feel-good sensations coming from your neurotransmitters. So we use antidepressants, selective serotonin reuptake inhibitors, block serotonin transporter only, prevent reuptake by presynaptic neuron. So we get more transmitted and less inhibited. And then we have some anxiety disorders that you can read about here. But a lot of times, our basic ideas with some of these mental disorders is we look at it as basically a chemical disorder. And the idea is that we have too much or too little of a certain neurotransmitter, or the neurotransmitter is being degraded too fast, or and if we can use the medication to try to control how that neurotransmitter is being sent or received by the neurons, then we can maybe improve your mood. Remember, these neurotransmitters are sending signals that are telling you, like, I feel good, I feel bad, I see things that, you can trick your brain based on how these neurotransmitters are sending and receiving signals.